I'm particularly honored to be here to deliver the Frank Dixon lecture in his honor. Anyone coming along in immunology at the time that I came along, and you've just heard that I've been around a long time, influenced in a big way by Frank Dixon. Frank Dixon was actually indirectly the reason that I started into immunology, but that's a longer story than I wish to tell now. What he symbolized to all of us and was drummed into Bob Good's trainees' heads was that he did everything in a very precise way. We were taught to make slides that looked like Frank Dixon's slides, with histological slides without any breaks or chatter marks that were perfectly adjusted to show exactly the point that he was trying to make. In fact, what one realized in watching Frank and being around him at meetings, even if not working with him, was that he had a very clear logic and an ability to cut through the fluff to the central issues at hand. And I'll mention him again in a later point in my talk, but he had a very strong influence on me and virtually everyone in the field of immunology. And for me, it was a great honor, as mentioned by Michael, to be offered the possibility to work with him. Unfortunately, I didn't quite ever make that possibility. So Michael has already indicated to you that I'm going to be considering how lymphocytes might have evolved. And when we began these studies, and we was Jan Klein in Tübingen at a Max Planck Institute, we were interested in trying to go back and trace the origin of our adaptive immune system. And we knew at that time that every living creature, even bacteria and single-celled eukaryotes, have immune defense mechanisms, but only in our vertebrate lineage are there lymphocytes that have, each of which has its own particular receptor, allowing it to see with a potential repertoire, an enormous repertoire, virtually anything, any pathogen, any bacteria, any virus, any other non-self or self component in existence. And to remember previous encounter. It was also known that we have basically the same kind of adaptive immune system, all jawed vertebrates, even the most primitive cartilaginous fish like sharks, skates, and rays. We all have a thymus where T cells are generated that Michael mentioned. We all have hematopoietic tissues where B cells are generated, variable, diversity, and joining genes that are put together in various combinations. And we have the enzymes that are important for that VDJ recombination, and then also activation-induced deaminase, or AID, which is important in generating class switch, which he mentioned, and also somatic hypermutation to make better fit antibodies, and also for, in birds, to generate by gene conversion a repertoire to begin with. So we set out to look at the not having any of these earlier jawed vertebrates, because we only know about them through their fossil remains. The only two living, more basal or primitive species were lampreys and hagfish. And lampreys had been said to be able to make agglutinins that were specific after immunization with, let's say, human blood group, blood cells, erythrocytes. So we went back to look at lampreys. Lampreys are worldwide in their distribution. There are about 40-something species of them. We study mainly the sea lamprey, and which has a complicated life cycle. They are predatory in their adult phase because they kill fish, and when they made their way into the Great Lakes of North America, they wiped out the native fish. And then they reproduce, fertilized eggs, and start the life cycle over again. We do most of our studies with these larvae when they're about 3 or 4 years old, about the age of an old mouse. If you cut one of those larvae in two and look at the inside tissues, they have a blood-forming tissue that runs the length of this very simple intestine, where all the different kinds of blood cells are made. And Jan and his colleagues had found a small round cell that looked like a lymphocyte that expressed a transcription factor, SPI-B, that has been shown in mice to be important in hematopoiesis and B lymphopoiesis in particular. Also, hemopoiesis occurs in this ventral aspect of these kidneys. They don't have lymph nodes or a true spleen as we do, or an obvious thymus. So our strategy was to take cells from these tissues that had the light scatter characteristics of lymphocytes that looked with blood stains, standard blood stains like lymphocytes, and even by EM. And to make an expression library, a cDNA library, to look at the genes that they were expressing. So we got what was high throughput sequencing in those days, annotated our catch from the sequences that were being expressed by comparing them with the known genes in the libraries. And we couldn't find any of these genes that are cardinal elements in our adaptive immune system in making T and B cells with diverse repertoires, or class 1 and class 2 major histocompatibility genes. After going back and stimulating these animals with a cocktail of mitogenic substances from plants and also live bacteria and foreign red blood cells, and going back and making a selective expression library by these larger activated lymphocytes, we got an enrichment in sequences that were had leucine-rich repeats. And we were not too happy about that, but we noted, and Zev Panzer had joined my laboratory at this point, that the first 22, which were a complete open reading frame, or almost complete, every one of these sequences was different from the other. They were different in that they had different numbers of these leucine-rich repeat modular units, and the ones that they shared also had different sequences, as you can tell from this color coding. As we took the longest one and just made a cartoon of it, they were like most of the, many of the leucine-rich repeat proteins. And as far as I can tell, every living thing on our planet uses this leucine-rich repeat modular unit in some way for cell-cell interactions and a variety of different functions. All of these that I've just been telling you about had a signal peptide, but an N-terminal LRR and a C-terminal. These are called capping peptides. They cap the ends of these solenoid structures which these sequences form and protects the hydrophobic core. In between, all of them had an LRR1 of about 18 and a connecting peptide of about 13 residues. The ones that were variable in number are these green blocks of 24 amino acids length for each of them. And they also had this invariant stalk region that was the same sequence for every one of these, even though this part was different in one versus the other. They also, they were rich in threonines and prolines and had 8 cysteines in this carboxy terminus hydrophobic region. They also had a predicted site that you could cleave them with a GPI, that they might be cleaved here and linked to the cell surface by phospholipase. And we expressed this one with a tag on it so that we could identify it. And in mouse thymocytes, and it came to the surface. And if we treated those cells with phospholipase C of bacterial origin, it cleaved these off of the surface. So it seemed to be that. This is a phylogenetic cartwheel diagram that just shows of the first 112 of these that we call them VLRs, for variable lymphocyte receptors, because they were expressed by lymphocytes and so variable, we assume that they were receptors. And what this shows is that almost all of them were different from each other. And in this cartwheel, where they are compared with the closest relative or closest sequence, these two, around 10%, back up 10%, are about 20% in sequence different from each other, and so on around this diagram. There were three, though, that were identical. And we weren't sure if this was an artifact of our polymerase chain reaction, or whether these were from stimulated larvae, so they could have been clonal or expansion as well. These asterisks that are included around here by some of these were where Zev did single-cell PCR and could only get one sequence, suggesting that only one allele was expressing these, or allelic exclusion, like our antibodies, except something very different. In trying to find out if these were expressed by a large number of genes, we couldn't find but one in a genomic library made from the sperm of a single lamprey, sea lamprey, that Chris Amamiya had made in Seattle. And in that one, we found this one VLR gene, and it was very incomplete. It only encoded for part of the N-terminal and two pieces of the C-terminal of this, and the stalk region, as well as the signal peptide. In between these pieces were large intervening sequences that were non-coding. But flanking, when we got genomic sequences, this gene, there were multiple LRR coding sequences that, if brought in and stitched in somehow with this, the intervening sequence deleted, could account for the kinds of genes that we were seeing, these VLR genes in their assembled form. And indeed, in lymphocytes, this process occurred to give you this smaller product without this long intervening sequences. And we sequenced dozens of these, and they too were all different, one from the other. Now, we don't know exactly, even now, just how this process works, but this diagram can tell you what we and others, and in particular Nagawa and his colleagues in Japan, have found. So there are hundreds of these neighboring segments or potential donor sequences flanking the incomplete VLR gene that I've been talking about, and this is a simplified version of it. And sequences that match in homology from a donor sequence can be brought in with complementarity to the recipient and then copied in bits and pieces. They can be copied from the start in the middle, stop along the way. So that process, though, occurs over and over and over until you get to the other end and fashion a gene for a complete VLR gene. The process can start from either end, and their studies, Nagawa and colleagues, suggested that it only began on one end or the other, not both, for this particular gene to result in a... So these serve as templates, these donor sequences. They're not moved from their original position during this process. And the genes that are thought maybe to be involved in this, I mentioned earlier, there's an AID, or activation-induced deaminase gene. This is a human version. It's a multi-exon gene. And from studies in mice and humans, this gene was discovered by Hanjo and his colleagues. And this is an essential enzyme for the initial class switching, and also for the point mutations that occur in affinity maturation in germinal centers. And it's also, in birds, has been shown to be essential for gene conversion for generating antibody diversity in birds in the first place. Lampreys, it turns out, Igor Rogozin and Zev Panzer and their colleagues found, have two of these lookalike genes, or orthologs, that they name cytosine deaminase A1 and 2. 2 looks more like our AID. 1 is smaller, has a single exon, and may have gotten into the genome by reverse transcriptase activity from this as a donor. That's a guess. I'll come back to those later in the talk. So at this time, this was the model that we deduced from this data, that in the jawless vertebrate, in the Lamprey gene, cells were assembled by using, in a random fashion, to be as donors to make these complete VLR genes, which were expressed on only one allele per lymphocyte. And somehow, to explain agglutinins that had been seen before, they must have multivalence in their secreted form, if that was the answer. Our colleagues at NCBI, Igor Rogozin and Lax Iyer, calculated from over 600 sequences that we provided them that the potential repertoire for these VLR-expressing lymphocytes was greater than 10 to the 14th. Probably much greater since it's used in bits and pieces. At least as great as our heavy and light chain B-cell repertoire. To set out on testing that idea, we immunized lamprey. This is injecting them in a coelomic cavity. This is an insulin syringe. You can see they're pretty small. After a first immunization, here we're injecting human O-positive erythrocytes. And after a couple of weeks, they all make antibodies in low titer. If you boost them by another injection, the level of those antibodies goes up about 20-fold. So, and we had made this stalk region, this invariant stalk region, we made a monoclonal antibody in mice that we could use to remove all of this agglutinating activity. So this shows that the antibodies that see this O-positive erythrocyte are these VLR antibodies. The cells that make them, and here you're looking in a cross-section of the gills in a lamprey larvae, and in the blood vessels they're loaded with these green cells that are making this VLR, which we call VLRB now. The blue stain is DNA. The red is the incorporation of BRDU by dividing cells. This is before and after immunization with anthrax exosporium. And you can see that yellow cells that are labeled with the red and the green are increased in number, indicating that they proliferate after immunization. The cells that secrete these agglutinins are cells that look like plasmablasts, as shown here, or mature plasma cells that I don't show you. Brandt Heron, by doing a Western blot with this anti-stalk region antibody, a monoclonal antibody made in mice, could show that the secreted VLRs were very large off the scale of this experiment, but reduction of disulfide bonds brought them down into smaller units. The variation in size is because of the reason I mentioned before. They have different numbers of these green blocks, or LRRVs, in them. It's hard to study, of course, these, so Brandt devised a way to make recombinant monoclonal antibodies after immunizing the larvae and following this. And one of them that was specific for this human O-positive erythrocyte antigen was used to look in a negative E image electron microscope image. And you can see that this cartoon shows you they come in pairs, 5 pairs. The stalk region where they're bound together is this density shown here. The stalks themselves are very flexible, so we saw many different kinds of shapes. But basically, this looks like an IgM antibody, except that it only has one kind of chain, and they're paired. By truncating the ends of this, the constructs encoding this, Brandt could show that they are tethered together here, and they are released as single units if the ends of the stalk region are truncated. Also, we can change the site direct. We can mutate these cysteines, and it takes at least 4 of the 8 that I mentioned earlier to tether them together in this multivalent form. Now, in Ian Wilson's laboratory here at Scripps, Byung-Woo Han was able to solve the structure between this part of the VLRB antibody, this RBC, red blood cell 36, with the H trisaccharide, which is the O-positive blood group antigen for humans. And here in this diagram, he shows the hydrogen bonds between 1 but in this LRRB2, 2 in this N or LRRBE, and a point that I've not mentioned, there's a highly variable insert in this carboxy terminal LRR that encodes this loop that also has important hydrophobic interactions with this H trisaccharide. One can see that more easily in a space-filling model. The gray is the H trisaccharide. And you can envision it by just thinking of a cupped, slightly bent hand with a thumb sticking out at the base of the hand, forming a little pocket in this site. And it's binding to these residues in these beta sheets in this palm of the hand. Now, these antibodies that Lampreys make are very specific. This one, again, is placed on a glass array of different carbohydrate epitopes or moieties, so that 600 and over 600 different carbohydrate radicals are put in spots on this glass slide by Rick Cummins, our colleague at Emory, who has a glycomic center. And as you can see, the reaction spikes that occur here, and here are the sequences. Every one of them has a fucose with an alpha-1,2 linkage to a galactose that has a beta-1,4 linkage to the next carbohydrate. And everything else doesn't react at all on this array. These are the most specific antibodies that have been seen yet to that particular determinant. Robert Kirchdorfer in Ian's laboratory again solved the structure for a VLR4 antibody that Brandt monoclonal that he made that reacts with a coat protein or a surface protein of the anthrax spore. It's very dense, and that's what mice see as well as the dominant antigen. And its too is highly specific. Here's another one made against this enzyme, hen-egg lysozyme, that's called HEL2D, that was made by Zev Panzer, and the structure was solved in Roy Mariuz's laboratory. And they showed 2 important points that I'd like to emphasize. This part, the LRR portion, is very rigid. It doesn't wiggle at all. That they could see, whereas this thumb has conformational adaptability, and it bound into this cleft in this enzyme. This is the catalytic site for hen-egg lysozyme. Heavy and light chain antibodies that are made by mice and humans, for example, mostly see these flat or curved epitopes out here. There are a couple of other examples where there are long loops from heavy chain only antibodies camels and sharks make that can do the same thing. So at this point, it looked as if there was a single line of lymphocytes in lampreys that had these VLR genes with these flanking sequences that could be put together in this fashion that I briefly described and expressed on the surface, and when the antigen fits the receptor on the surface, it can stimulate them to enlarge, divide, differentiate into cells that secrete the antibodies. And I thought, aha, that shows that B cells came before T cells. That turned out not to be the case. One of the many errors that I've made in this line of research, it turns out that lampreys have 3 VLR genes. We had found with Kasahara, who was studying hagfish, that there were 2 of these VLR-like genes. And Kasahara and his colleagues, after finding a second one in lampreys, Kasahara found a third one, and these are called A, B, and C. You can see that all of them are incomplete. They have intervening sequences. These are not as lengthy. These are not according to scale for A and C as for B. And they all have invariant stalk regions. And each of these sequences for these A, B, and C differ from each other. So Pongoa and Masa Harano in our group could use these to make monoclonal antibodies that would discriminate between these 3. And when they stained lymphocytes from these lampreys, they could show VLRB cells are different from A's, C's are different from A's, C's are different from B's. So they all were on different populations of lymphocytes. They expressed either A, B, or C. When we injected these lamprey with a classic T cell mitogen that induces them to divide, we found that the VLRAs proliferated. Here we're looking 9 days later, and the VLRCs also proliferated. The VLRBs didn't proliferate much at all, as shown by uptake of this DNA precursor EDU. So these A's and C's began to look a little bit like T cells. And you remember I told you that there were 2 of these cytidine deaminases, CDA1 and 2. When we did expression by these different populations, A positive, B positive, C positive, and triple negatives that didn't have any of these markers, didn't react with any of these antibodies, we could see that the CDA1 was preferentially expressed at higher, let's see if I can, not sure how that happened, but okay, I think I'm okay, thank you. And not by VLRBs, whereas the VLRBs preferentially express the CDA2. VLRBs also express transcription factors that we know to be essential for B cell differentiation and function in mice and humans. And whereas the VLRAs and Cs express some genes in common that are involved in T cell differentiation and also by progenitors that migrate to the thymus, activation elements that are involved in signaling, a cytokine receptor, a cytokine, and another interleukin that's an important inflammatory cytokine. So I'll come back to these other aspects in just a moment. So they're beginning to look as if these might be like T and B cell or prototypic T and B cell, and neither of them secrete their products, their receptors, as the B cells do when they differentiate. They both respond to PHA stimulation that I mentioned before. I don't show you the data, but they also preferentially respond to cells from other lampreys, alloantigens. And whereas VLRBs respond similarly to every kind of antigen, from a mouse or a frog as well as. So these tend to see allodeterminants, much like T cells see MHC. One of the questions, of many questions that's raised, is where are these receptors assembled? Where are they put together? And is there a thymus equivalent where T cells are generated in mice and men? Well, the site that turned out to be where they seem to be assembled is in this gill region, in these gill folds. I mentioned that CDA1 is only expressed in the T-like cells, and it's expressed only in the tips of these gill folds. They're cut in a cross section. And this first of these secondary filaments, right out at the end in these blue areas, is whereby in situ the CDA1, which is only expressed by this type of cell, this expression wasn't changed regardless of how we stimulated the lampreys. If we injected them with this T cell mitogen, PHA, or if we immunized them with anthrax and other antigens, this staining pattern or this expression pattern doesn't change at all. It seems to be constitutive. Also, this is a gene that's expressed by epithelial cells in this area, FOXN1. It's the gene that's mutated in nude mice that don't form a thymus or T cells, and the same is in nude humans with that mutation in this gene. This is also cells in this area as well as elsewhere. Some of them express the VLR, the receptor genes themselves, and we could show by monoclonal antibody that there were cells expressing the protein here out in this, we called it a thymoid area, which has both lymphocytes and epithelial cells in that location. Our collaborator, Thomas Boone and Baji, used laser capture microdissection to get DNA samples from this area that we're calling the thymoid. And when they did sequences of these DNA, VLRAs in this case, almost 30% of them were non-productively assembled. That is, they had a stop codon in their sequence or a deletion or something else that prevented them from being a productive assembly. Out in the bloodstream, down below here in blood vessels, none of these, all of them had productive assemblies, suggesting that these were somehow selected in that area for productive assembly. VLRC is the same. And if they used laser capture microdissection to pull out DNA samples, again, frequently in this thymoid area were non-functional assemblies. Some also were found in the blood for the VLRC, but much less than in the thymoid area. Now, some of these were actually incomplete assemblies, which we had seen before, but very rarely in other places. And for the VLRC donor sequences, and we've found about 180 sabidas in our laboratory, potential donor sequences, and all of them come in these flavors, as you can see in these cartoons. And that tells you how, if you have to have sequence similarity between this and what's in the germline incomplete gene to start with, this one has to come first. Then this one has to come to complete that LRR1. Then one of these has to come to complete this LRRV and so on. And the same, you can tell how you would have to start on this end with using only these kinds of 5 types of building blocks. The point here is that these are incomplete assemblies that are found enriched in this thymoid region, suggesting that this is where the assembly is occurring, and it can occur beginning on either end in this site. Now, these are sort of, this is what Bob Good would have called an oh hell slide, one that you couldn't make out what it actually means. But it tells, there are a few points that I'd like to make. It's the same. Here's the incomplete gene, and there are large blocks that are close, that resemble each other very closely. They have the same constituents, spaced in the same way, and their sequences are almost the same, suggesting that one of these duplicated to give rise to the other. There are smaller block duplicates that give you some hint about how this diversity is generated. And it's also a point that I'd like to make is for this VLRC, there are only 2 potential donor sequences that can come in here and change this germline configuration. And they can't change it very much because they are very close in sequence to each other. And they are not very different from the original sequence in the germline gene that needs to be completed. And and they don't have a sequence that would code that loop that I showed you for the thumb-like region. So they must see whatever they see a little differently. Now, because of the size difference in this intervening sequence, you can tell the germline gene from the assembled gene. And I won't go through all of this, another oh hell slide, but Masa Hirano could gather up lymphocytes from around in the body of these lamprey larvae. And several points that I can make. These 2 VLRA and C incomplete germline genes are located close to each other. VLRB is on the same chromosome, and it's quite a distance away. So it's not close to these. When I told you that this one was expressed on one allele or the other, and when if you look at VLRB expressing cells, they have they've assembled and expressed VLRB transcripts, but they don't do anything in this VLRC locus or this VLRA locus. Conversely, the VLRAs actually positive cells not only have assembly in the VLRA locus, but also something is happening in this bulk sample in the VLRC locus. But nothing is happening in the VLRB locus. So without going into a lot about that slide and with many single cell analyses of the receptors, this is the model that the data that Masa has assembled suggesting. So it suggests that a progenitor cell comes into this thymoid region and first attempts to get a productive assembly on one allele. If it fails, then it has another try on the other allele, which would allow it to survive. If it misses on both alleles, it's still got another chance because it can go to the VLRA. And if that works, then it can be salvaged as a VLRA expressing lymphocyte. For the VLRAs, which seem to begin later, first one allele, if it works, fine. If it doesn't, there's another chance, but nothing, it's not going to make it after that. Now, I mentioned too that there were many similarities in the gene expression profile for these VLRAs and VLRCs, but there's some striking differences in this limited gene profile of orthologs or lookalikes of genes that have been defined in mammals. This TCF1 gene in mice is essential for alpha-beta T-cell differentiation. SOX13 is another transcription factor that inhibits this TCF1 transcription factor, and at least in mice, it can block alpha-beta differentiation among its roles and is essential for gamma delta, and it's only expressed, T cell development is only expressed by these VLRCs. There's some other genes that are preferentially expressed by these VLRCs, and I note particular, these are homing or integrants that decide where these cells, where gamma delta cells in mice can home to the epithelium. When we look to see where these cells were going in the lampreys, and here the staining of the VLRC expressing lymphocytes is green and red for VLRAs. In this kidney, where there are lots of hematopoietic cells and lymphocytes, you can see that the greens and reds are just about equally mixed. In the epithelium of the intestine that's shown here, this being the kidney, this is the intestine, there are more greens than reds, but not a huge excess, and that's shown here. But in the skin, only in this epidermal level, the most outermost level of the skin of the lamprey larvae, you can see that they're mainly all greens, and there's a large preponderance of these green cells, much like gamma delta cells in Wendy Haverin's studies here at Scripps. Now, in mice that she studies, these T cells that go to the epithelium all express one kind of T cell receptor that she's engaged in finding out what it sees. We're not stripping off just the epithelium, but both dermis and epidermis here. But when Massa did sequencing of 83 of these VLRC assembled genes, you can see that there are 22 repeats of the identical sequence, 7 of this blue and so forth. So it's an oligoclonal distribution, which wasn't seen for these VLRCs in blood or kidney. And for VLRAs, it wasn't seen in any of these tissues. So again, these two lineages of T-like cells in lampreys are beginning to look like gamma delta and alpha beta T cells. Not exact lookalikes, of course, but some rather striking differences. Now, back when Frank Dixon and Bob Good and Dan Campbell and also Henry Kunkel formed a society that they called the Hagfish Club. I don't know if you ever heard of that before, but hagfish were considered, they live deep in the sea, feed on dead carrion, are mean. If you make them angry or stir them up, they secrete a slimy material that makes them hard to work with, and were thought to be immunologically null. So Frank, Bob, and Dan and Henry Kunkel formed this club they called the Hagfish Club. And they vied with each other for some outrageous act that they could, or accomplishment, to be the president of the Hagfish Club. And we wanted to know since hagfish are the sister group and thought to be even more primitive than these lampreys, if they have the same kind of immune system that lampreys do. And indeed, they have 3 incomplete VLRA, B-like, and C-like genes. I mentioned that we had found 2 of them, and Jianshu Li found a 3rd, and the VLRCs in both the hagfish and the lampreys are different in that they have fewer of these green blocks, so they a shorter insert in this carboxy terminus, excuse me. So they don't have this thumb-like region, neither of them. Instead, they have another loop here, but I won't go into that at the present time. So what this suggests is that both hagfish and lampreys, although we don't know as much about the system in the hagfish, they had a common ancestor about 480 million years ago, it's estimated. So this kind of unusual type of receptor system that involved these VLRA, B, and C genes being assembled in the incompletely understood fashion that I've mentioned here, instead of generating antibody B cell and T cell receptors using RAG1 and RAG2. In fact, there must have been a common ancestor, since both have AID-like, both jawless and jawed vertebrates have an activation-induced deaminase-like apobac family member. That gene must have evolved in a common ancestor. And in fact, since these prototypic two major T-cell lines are present in both jawless and jawed vertebrates, as well as a B-cell-like line, that implies that this genetic program for these types of lymphoid cells, T and B cells, was present already in a common ancestor of these somewhere around 500 million years ago. And that asks more questions than answers being given by it. We don't know what their receptors might be, and so forth. I'd like to just close, though, in the last few minutes to consider another question. We've known for some time that there are cells that don't have lymphocytes, that don't have T and B cell receptors, natural killer cells that have natural killing cytotoxic activity against virus-infected cells, tumor cells, and so forth. And in the last 5 years, we've learned that there are innate lymphoid cells, or ILC cells, that we hadn't appreciated, that immunologists hadn't appreciated before. These innate lymphoid cells are sister groups to the natural killer cells, and they come in 3 different or 4 different types, depending on how you classify them, called ILC 1, 2, and 3, and this other one called a lymphoid tissue inducer that's ILC3-like, in that they use some of the same transcription factors in their differentiation program. These were missed for a long time by immunologists because they are very scarce, they are not abundant, and they are not found in any frequency at all, or in very dwindling frequencies in the blood, or in lymph nodes. They are found mostly in lymphoid tissues like the intestine, lungs, and located beneath barrier surfaces. So we're just beginning to learn more and more about them, and they require these different transcriptional factors to... So in asking if lampreys, when these evolved, we thought, well, we could go back and look in lampreys. I mentioned these triple negative cells because the NK cells and ILCs shouldn't have these receptors. So if in red here are the frequency of cells in these different tissues that don't have, or triple negative for these VLRA, B, and C. Now, you've noticed that in these little limited gene expression profiles that I've been showing, that in the triple negatives, it's absolutely blue or white. There's just nothing that we were seeing as far as any of these genes being expressed preferentially. But I've already alluded to the fact that there are transcription factors that are essential for differentiation into the NK cells, into the progenitors. This ID2 is essential for development of ILC 1, 2, and 3, and also the lymphoid tissue inducers, and so forth for these other transcription factors that are important to the program for these different pathways. And if you look at some of these genes in this triple negative fashion, here's ID2, PLZF, infill, some of the same transcription factors that are used by these innate lymphoid cells or NK cells. This is a gene that is for, this is the NCAM1. So I guess that's appropriate for this neural cell adhesion molecule 1 that we were in the auditorium of the discoverer of this gene a long time ago. It's expressed not only on neural cells as an adhesion molecule, but on NK cells. And these genes are also expressed by NK cells. So in going back and looking, NK cells have these granules, acidic vesicles. And there's a marker, a fluorochrome marker that's taken up and activated in acidic vessels, cells that have acidic vesicles. And you can see in these triple negative cells, and I'm sorry it's not really labeled properly here, there's a population that's not a VLRB or VLRC cell that is labeled with this lysotracker. And here's the control. Monocytes, they are not. So if you take all of these and with neutrophil markers as well and strip them out, you can begin to dissect this triple negative population and then look to see about transcription of these different genes. So here's the lysotracker positive, but negative for A, B, C, monocyte, and granulocyte markers. And this ID2 gene is not expressed by them, whereas the other fraction of these lysotracker negative, pentanegative, not having any of these markers, expresses it. Same here, but this is the other way around. So lysotracker seems to mark a subpopulation of these VLRB negative or VLR negatives. And this is just a summary of the profile. So the lysotracker positive express several genes or surface molecules that are expressed by NK cells, whereas the non-NK, like the ILCs, tend to express these transcription factors that are important. So we could pull out one of the markers that we could use. John-Shu Lee has made a mouse monoclonal antibody against a Lamprey TLR5. That's a toll-like receptor that's expressed by other types of cells, by NK cells in humans. And it's expressed at very high levels in different larvae, this one being the oldest, and if it allows you to pull out these cells, and this, you could take this EM picture and match it with a human or a mouse NK cell, natural killer cell, and it would look exactly the same. It has these short, thin microvilli. It's not a smooth surface. It has a lot of cytoplasm. The nucleus is off to one side of the cell. The heterochromatin is stretched around the nuclear margin. And inside, there are lots of these granular vesicles that have varying amounts of this electron-dense material. So to putting that all, we've not shown that those are actually natural killer cells yet, but they look like natural killer cells. They're expressing some of the transcription factors that are needed by NK cells that have been defined in mammals. And many of the genes that are in the program that's necessary for this ILC2 pattern are present, suggesting to us, well, to conclude, I can't come to many totally solid clues, but two major points. One is that T and B cells, which we've only known about for about 50 years, have been present for at least around 500 million years, and they've been constant companions over that period of time. The reason that they've been selected and evolved for different functions is speculative, but presumably because they are work together in protecting against infections cancers perhaps, and have survival advantage. The sketchy data that I showed you in the end, even more sketchy data, suggests that the basic genetic programs for innate lymphoid cells probably evolved around the same time or may have started in their evolution even earlier. And I'll like to just point out that the people whose names that I've mentioned, Sabi, Masa, Brandt, John Shu, Yoichi, are shown here, who did the work, and our work has been, we've been aided by many different people. I mentioned people here at Scripps and others we're working with now. And our work has been supported by, of course, National Institute of NIH, Georgia Research Alliance, and these are colleagues who've created the environment that we function in. And with that, I thank you for sticking through this long talk and honoring Frank Dixon, and again express my appreciation for being able to participate in this occasion. Thank you.